Today, we're going to dive into this incredible, elegant cellular dance, the one that creates the blueprint for life itself. Let's jump right in.
Okay, so have you ever really stopped to think about it? I mean, out of the billions of people on this planet, there is absolutely no one else exactly like you. That's not a fluke. It's not an accident. It's totally by design. And the answer to why? Well, it's all down to a process your body perfected eons ago. And that process, it's called meiosis. Now, this isn't just any old cell division. It's a super specialized form that's all about making gametes, sperm and egg cells. You can think of it like biology's ultimate card shuffle. It's the engine that creates all the amazing genetic diversity we see everywhere. All right, here's our game plan. First, we're going to look at the big picture, life's blueprint for you. Then we'll break down the two main acts, Meiosis I, the Reductional Division, and Meiosis II, the Equational Division. After that, we'll touch on what happens when things go wrong. Errors in the blueprint. And finally, we'll tie it all together and see why this whole thing matters so much.
Okay, let's kick things off with the master plan. What is meiosis actually trying to do here? What's the end goal?
So the mission, it's pretty straightforward, but it is absolutely crucial. You start with one single diploid cell. Diploid just means it has two copies of every chromosome, right? One set from your mom, one from your dad. Then this one cell goes through two really precise rounds of division. And the result? You end up with 4 haploid cells. And haploid means they've only got one copy of each chromosome. But here's the kicker. None of those 4 cells are identical. They're all unique.
So let's get a bird's eye view of these two big stages. First up is Meiosis I. We call this the Reductional Division. Its whole job is to separate the homologous chromosome pairs. The matching chromosome from mom and the one from dad. This is the step that literally cuts the chromosome number in half. Then you've got Meiosis II, the Equational Division. This one is a lot more like regular cell division. Its job is to separate the identical sister chromatids. All right, let's dive deep into that first phase, Meiosis I. Buckle up, because this is where the real magic happens. This is where all the most important genetic shuffling goes down.
Now, before we even get to the step-by-step, we have to talk about this absolute superstar of a process, crossing over. I mean, this is incredible. It's when those homologous chromosomes, the one from your mom and the one from your dad, they get super close. They literally hug, and they swap pieces of their DNA. They're creating brand new combinations of genes that have never existed before. This is like a prime source of all genetic variation.
Okay, so here's the play-by-play. It starts with prophase I. The chromosomes pair up and do that crossing over thing we just talked about. Then comes metaphase I, and this is where another cool thing happens, independent assortment. Just picture it. All those pairs of chromosomes line up right down the middle of the cell. And for every single pair, it's a complete 50-50 chance, a coin flip, whether the mom chromosome or the dad chromosome faces one way or the other. This random lineup is another massive source of genetic mixing. After that, in Anaphase I and Telophase I, the cell just pulls those pairs apart and splits in two.
And what's the results? Well, after just that one round, we've already got 2 haploid cells. And thanks to all that crossing over and independent assortment, they are totally, completely unique, different from each other, and different from the cell we started with. But we're not done yet. The job's only half finished.
Which brings us to Act 2, Meiosis II, the grand finale. This part is all about one thing, separating the 2 identical copies, the sister chromatids, that make up each chromosome.
So those 2 new cells we just made, they basically go through the whole process again, but with one really important difference. In Metaphase II, the chromosomes don't line up in pairs. They line up single file, right down the middle. Then, in Anaphase II, the cell doesn't pull pairs apart. Instead, it pulls apart the sister chromatids, those 2 identical halves of each chromosome. Then, the cells divide one final time. And the final result? 4. That's it. From that one single cell we started with, the process has created 4 completely unique haploid cells. 4 potential blueprints for a brand new individual. Pretty amazing, right? But, this whole thing is so complex, so intricate. What happens when it goes wrong? 'Cause let's be honest, with this many moving parts, mistakes, they can happen. Errors can creep into the blueprint. And those mistakes can lead to what we call chromosomal abnormalities. That just means that the sperm or egg cells that get made might end up with the wrong number of chromosomes, or the chromosomes themselves might be, well, broken.
or put together incorrectly. So the most common issue is having the wrong number of chromosomes, and this usually happens in one of two ways. First, you've got nondisjunction. That's a fancy word for when the chromosomes just fail to separate correctly during anaphase, that pulling apart stage. Or you can have what's called anaphase lag, which is pretty much what it sounds like. A chromosome just lags behind, gets left out, and doesn't get included in the new cell.
And the consequences of these kinds of errors can be really significant. They can lead to genetic conditions like Down syndrome, which happens when there's an extra copy of chromosome 21. We call that Trisomy 21. Other examples are Edwards syndrome and Patau syndrome, which are caused by extra copies of chromosomes 18 and 13.
It's not just about having the wrong number, though. You can also have errors in the structure of the chromosome itself. A classic example is translocation. This is where a chunk of one chromosome literally breaks off and gets stuck onto a totally different chromosome. This kind of major rearrangement can really mess with important genes and lead to all sorts of problems. Looking at what can go wrong really drives home just how important it is for this process to go right. So let's pull back and wrap this up by looking at the big question. Why is meiosis so incredibly fundamental to life as we know it? At its core, it's this. Meiosis is the absolute foundation of sexual reproduction. It's the engine that powers genetic variation, and it's that variation that allows species to adapt, to change, and to evolve over millions of years.
So, what are the big takeaways here? If you remember anything, remember this. 1. Meiosis makes those special haploid cells you need for sexual reproduction. 2. It cuts the chromosome number in half, which is crucial for keeping it stable from one generation to the next. 3. It is a master at creating genetic diversity through things like crossing over and independent assortment. And 4. All that diversity, that's the raw material that evolution has to work with.
And that brings us to one last big thought. We've seen how meiosis is this beautiful, almost impossibly intricate dance that makes every single one of us unique and gives life itself the ability to adapt. It truly is life's engine for variation. So the question I want to leave you with is this. In a world that is changing faster than ever, what does that engine, what does that variation mean for our future? For the future of our species and for all life on this planet?